Hello students, this is Mr. Russell. I'm here to talk to you about experimental design, which is something you've heard about, but it's really important to all science because that's what science does, is experiment. So we need to make sure we are always improving our understanding of experimental design. And I'm going to use an example from my summer in order to try to explain that. So in June, I guess it was, I was barbecuing in my backyard. It was after dark, and I needed to make an adjustment to my smoker because it was either too hot or too cold. I don't remember which. But it was dark. I couldn't see what I needed to do, so I foolishly, don't ever do this, used a blowtorch for light instead of a flashlight. The flashlight, by the way, was only about 10 feet away. This was very foolish. Don't do it. Anyway, I used the flashlight. I made my adjustment. I turned, I'm not sorry, not the flashlight. I used the blowtorch. I made my adjustment. I turned the light off. And then after it was out, it was still very hot, and I accidentally touched it to my arm, and you can see this little dark spot right here. Pretty nasty burn that I got, blistered up almost immediately. My wife was there, and her initial reaction, of course, was to laugh hysterically because this was so dumb. After she stopped laughing a few minutes later, she first of all got mad at me for doing something so foolish, and secondly, she told me to go put some Neosporin on. Okay, Neosporin, I'm sure you've heard of, and there's a lot of other kinds. Neosporin is just one brand, but it's stuff you put on cuts or scratches, and it's supposed to keep germs from growing. And that's good for three reasons. A, most importantly, it will hopefully prevent infection, because infections can cause things to heal slowly, and they also, sometimes if they get really out of hand, they can kill you. It also will help it heal faster for various reasons, including because it holds off infection. Third one is it's supposed to, I don't know if this is actually true or not, but it's supposed to prevent scarring while it's healing. Mine has been there for a couple months, and there's definitely a scar there. We'll see if it lightens up as time goes on. Anyway, but I want to use this example to talk to you about experiments, because I was thinking, I wonder if that stuff actually works. I wonder if it actually kills germs. So I decided, I'll set up an experiment. Okay? So let's go through this. What is an experiment? An experiment is an ordered investigation attempting to answer a question. Okay? The universe is full of questions. Why is the sky blue? Why do my feet stink? Wow. Why does Mr. Russell do foolish things like use blowtorches for flashlights? Lots of questions out there. And there's lots of ways to answer them, but in an experiment, we have to follow a certain order of things, okay? We do things in a certain order and in a certain way. And that makes it so we can trust the results that we get at the end of the experiment, okay? So in this case, I want to know, is the Neosporin actually killing germs? Maybe you're a farmer growing plants and you want to know which fertilizer works better, okay? Those would all be examples of that. And that takes us to our next thing, which is observations, which is the first part of it. So, and this is really where that light bulb kind of goes off in your head. Maybe it's not a light bulb, maybe it's a question mark that says, I wonder why, ... I wonder if Neosporin actually works. I wonder if my tomatoes aren't growing well because last year I used a different fertilizer than this year. I wonder if my feet stink because I smear butter on them before I put my shoes on in the morning. I don't know. Okay, these are observations. Okay, and that's where we start out with. This is really just the curiosity phase. This is wondering. Okay, the next phase, I'm sure you've heard of this, is a hypothesis. You've heard before that a hypothesis is an educated guess. That's really not the way we look at it in high school science classes. A hypothesis is a prediction, okay? So if we set up an experiment, what is going to happen? What do we think is going to happen? Okay, and a very common thing here I want to point out is that people think their hypothesis has to be correct. They're going to get a bad grade if the experiment doesn't support their hypothesis, and that is not how science works. Thomas Edison had some fantastic quote about how when designing the light bulb, he didn't fail 10,000 times, he just found 10,000 ways that didn't work. And then what did he do? He built that light bulb. Okay, so a hypothesis is a prediction, all right? So I think if I use last year's fertilizer, it will grow better than this year, okay? Or my tomatoes will grow better than this year, all right? That's a hypothesis, a prediction. A lot of times we put it into an if-then statement. If I do this, then this will happen. If I use last year's fertilizer, then the tomatoes will grow better. If I put Neosporin on germs, then they won't grow. If then. Okay, so let's talk about more parts of an experiment. All right, what are we testing the effect of? There's something in this experiment and we want to know if it is doing something. Okay, so in the case of this flesh wound, you want to know, is the Neosporin actually killing germs? Okay, so what are we testing? The Neosporin. All right, that's our independent variable. We call it an IV for short. That's not intravenous, that's independent variable. Okay, so with our tomato plants, you think it's the fertilizer that's causing them to not grow so well. So you're like, I think that if I change the fertilizer, that will help. Well, then your independent variable is your fertilizer. It's the thing that you're changing. Okay. Now, in order to test if that independent variable is having some effect, we need something else we can look at, something we can measure or observe. If you're doing tomato plants, what are you going to measure or observe? You're probably going to come back a few weeks or a couple months later and see which ones are taller, or maybe you're going to count the number of tomatoes you get, or maybe see how big the tomatoes are. You're basically going to test and see how well these plants are growing. In the case of the Neosporin, we're going to see how well that bacteria and mold, those microorganisms, are growing, because that's what the Neosporin is supposed to be killing. So in order to test to see if this, the IV, works, we need to look at growth. Okay, so let's talk about how we're going to set this experiment real quick here. First of all, I'm going to use two of these. These are called Petri dishes, named after a guy named Petri. And what they are is shallow dishes, and you fill them up with agarose, which is something similar to gelatin in consistency, but it's made out of seaweed, and it's a great medium or substance for microorganisms to grow on. It also has nutrients in it that they need to grow: fats, proteins, carbohydrates. It's heaven for them. It's like a total smorgasbord. If we put them in here, they're going to go nuts unless we do something else that keeps them from growing. Okay, so the way I have this set up, I am going to use these. This is swabs. If you've ever gone to the doctor thinking you had strep and he used a real long Q-tip and he swabbed your throat and then put it in a little plastic bag or something like that, he sent it to a lab and then they smeared it on one of these. Okay, and then they threw it in one of these, an incubator, which keeps it nice and warm, perfect growing temperature, and a couple of days later you come back and look, and if you see the right kind of little patterns growing on your dishes, well then you know that that's strep. We're not looking for strep, we're just going to see if the stuff grows in here. So what I'm going to do is put bacteria on these, and then I am going to put Neosporin on one of them to see what happens. Since the injury was on my arm, I am going to just sample my arm, which will be real easy. I'm going to take both of these, same time, same spot where this burn was, and just wipe them on my arm. Now this doesn't seem very germy and gross because, believe it or not, I did shower today, but your skin is just crawling with nasty, nasty stuff. So I'm going to take one of them and smear it on one dish. I'm going to twirl while I do that to try to get as much off of there as possible. I'm going to take the lid and put it on that, and then I'm going to take the other dish and do the same thing. Okay. All right, these have now been what in a biology lab they like to call inoculated, which means when you expose it to some living thing. Okay, the last step of the process here is to apply some Neosporin to one of these things. Okay, you'll notice that I've drawn a line down the middle of these and with the N on one side, so I only want to get the Neosporin on that side. Okay, so I'm going to spin that around. I'll even tilt this down so you can watch. Okay, I'm going to cover up the other side and then give this a real quick spritz and then cover it up. Okay, these will now go in to the incubator. which I have set at about 35 degrees Celsius. That's somewhere between 90 and 95 degrees Fahrenheit, and that nice warm temperature is going to be good for both mold and bacteria to grow. Okay, so let's keep looking at this. So, did our IV, our Neosporin, actually cause a change? The growth that we're going to see, or hopefully see, maybe we won't see. How do we know that it's what caused that? And that's why we have separate groups. You'll notice I had a dish labeled C, and I had one with the label N on it for Neosporin. C is a control group, and a control group does not get your IV. And that's so that if we just sprayed Neosporin on one and took it out and there wasn't anything growing on it, could we know for sure that it was the Neosporin that killed those germs? Who knows? Maybe my incubator shut off and started refrigerating and it just froze them all. What you want to have is a control group, which is a group that does not get the independent variable, and that is how you know that it must have been your IV that caused any change in your DV. Okay, and your Neosporin group is your experimental group. It's the one we're experimenting on. Okay, what are some other things that could affect the DV? What are some other things that could affect that bacterial growth in there? Temperature, the amount of moisture that they have, any other toxins that could have been produced. And that's why I put both of them in the same Petri dish. They have the same nutrient medium that they're growing on. They're going to be in the same incubator, so they get the same temperature. Those are all things that we hold constant because we want to know for sure that they didn't affect the dependent variable between the two groups. So you want to keep as many things the same as you possibly can in an experiment, and those are constants.